Hello and welcome to Nikolay's genetics lessons, and today I have prepared 3 slides for you, so even if you know the answer to the first question, please wait for the other 2, and one of them would be really hard and you are going to learn something new today 100%. So here is a pedigree. Which mode of inheritance best explains the pedigree above? Non-genetic, we usually use this explanation when we do not see pattern which explains some of this popular mode of inheritance of genetic disease. Here I see a pattern, so this is not variant A. Let's check other variants. Why it is not mitochondrial? Because if you take a look, if mother has mitochondrial genetic disorder, 100% of her children also should have this genetic disorder. But we see here, out of 3 children, 1 doesn't have. Here's another example. Mother has this genetic disorder and her daughter doesn't. Here's another example. So 100%, this is not mitochondrial genetic disorder. Next variant, X-linked dominant. Why it is not X-linked dominant? Because this male here, if his X chromosome is defective, he cannot pass it to his son. He only can pass Y chromosome. So there is no way that his son would be affected with X-linked dominant genetic disorder. Now we have 2 variants left, autosomal dominant and autosomal recessive. Why it is not autosomal recessive? Because in autosomal recessive, we see that genetic disorder may skip generations and then reappear. So we can see it, for example, in generation 1, we do not see it in generation 2, and then it can reappear in generation 3. Here we see that only those people who affected have affected children, and those who are not affected don't have affected children unless they make a couple with affected person. So the best explanation that this is autosomal dominant genetic disorder, and for example this person here has dominant allele A, which is defective, normal allele A, and his spouse has 2 normal A alleles. Both daughter got dominant allele from the father, and their son also got the same allele from the father, and from the mother they only can get recessive allele A, so they would be affected. And here genotype of this person is normal. This person can be heterozygous and affected, and the daughter here got dominant allele A from the mother side, recessive from the father, dominant allele A from the mother, recessive from the father, and their son got recessive allele A from the father and recessive allele from the mother, and his phenotype is normal. And the last couple here, we assume that this male here is phenotypically and genotypically normal, but their son got dominant allele from the mother, he is affected, recessive allele from the father, and daughter got recessive allele from the mother side and recessive allele from the father side, and she is phenotypically and genotypically normal. Theoretically, we also can explain this with autosomal recessive genetic disorder, but autosomal dominant would be the best explanation, would be the best choice. Here is the next slide in question. We have the same pedigree, and here's a question. What is the probability that unknown genotype-phenotype person in gray with arrow will go on to develop the condition? So we are talking about this person. We already have found that this is autosomal dominant genetic disorder, and genotype of this person have to be heterozygous. This person also have to be heterozygous, and genotypes of their parents have to be as follows. So homozygous recessive here, homozygous recessive here, and heterozygous here. Some of you may ask, but what if these affected persons are not heterozygous but homozygous dominant? They're still going to be affected, but this is not going to change genotypes of their children, no matter whether this person may have two dominant alleles. In this case, 100% of his children are going to be heterozygous, would be affected. But in this family, we see that one child is unaffected, and we know for sure that this female is heterozygous, because this person should get normal allele from the mother side and another normal allele from the father side. So here we know genotypes for sure. Here we are not sure. It can be heterozygous or homozygous dominant. Still, it's not going to affect our calculations. When we have two parents who are heterozygous, we can build simple Punnett square, and we can find probability for their children also to be affected. Take a look. So here is two dominant alleles, dominant and recessive, dominant and recessive, and two recessive alleles. This genotype and phenotype means normal phenotype, and this going to be affected. So 3/4 of the children of this couple are going to be affected, and 1/4 is going to be unaffected. So we have found that probability for child shown here with arrow in gray to develop this genetic disorder, the probability is going to be 75%. And the last question is based on the previous pedigrees, but we don't need those pedigrees to be shown here. We just have found that those pedigrees can be best explained with autosomal dominant genetic disorder. So here's a question. The affected people get their DNA sequenced, and the report identifies the following genes having variants of unknown significance. If you had to predict based on the inheritance pattern only, which two of these changes would be likely to be associated with the disease? Note that all are possible. This question is to think about what each of these mutations might mean for protein function. Variant A, a truncation mutation to the 3' end untranslated region of a gene involved in the extracellular matrix production. Imagine that after gene transcription, we got this messenger RNA, and it have to undergo certain post-transcription modifications, and here we have 5' end UTR untranslated region, and here we have 3' end UTR also untranslated region, but these regions are important for regulation of the protein translation from this messenger RNA. Only red part would be translated into protein, and this is going to be regulatory sequences. For example, 5' cap and 3' end poly A tail. As I said, poly A tail is basically just addition of the multiple adenines here, and the more such adenines would be added here, the longer life expectancy of this messenger RNA. So the longer protein product would be produced. Though this type of mutation can be both dominant and recessive, but in most cases it's going to be recessive and not dominant, because normal protein would be produced, but maybe in smaller quantities. And again, regulatory mechanism of the cell may also produce more RNA, thus it's going to be less stable in order to compensate for this unstable messenger RNA. Next question, a missense mutation affecting the DNA binding site of transcription factor. So here is a double-stranded DNA, and here I will show with the same color, but just want to separate from the gene coding region. This is going to be upstream region of the gene, and here we may have DNA binding site of transcription factors which help to transcribe a gene. And if, for example, here we have a sequence A, T, G, and due to single point mutation, now we have different sequence here, for example, C, T, G. And when protein binds to this sequence, now this change may lead that it may bind stronger or weaker. If it would bind stronger, probably this may lead to stronger expression of the gene. In other words, we can get here a gain of function. So this is going to be our choice, but let's check other three. A nonsense stop codon mutation in structural protein. Take a look. For example, normally we have stop codon, let's say somewhere here, but what is going to happen if stop codon is going to happen somewhere here? In this case, we are going to get small protein, and which is not going to be functional. So this is going to be a loss of function mutation. So this is going to be not our choice, because this is going to be usually recessive mutation and not dominant. If someone may produce normal messenger RNA this size, we say this is going to be normal allele A. And if someone would produce smaller size, we say that this is going to be non-functional small a. And only person who has two such non-functional alleles would express genetic disorder. But this genotype usually means normal phenotype, because one allele can express normal protein and another one is going to be non-functional, and still our organism would be functional because this normal protein, normal allele would be overexpressed in order to compensate this non-functional, and our organs still are going to have normal level of the necessary protein. One more time, nonsense or stop codon mutation usually means recessive genetic disorder. Next, variant D, a single amino acid change in a part of a protein involved in polymerization. So we are looking for changes that will exert dominant effect. And here, a change in polymerization might mean the mutant protein poisons non-mutant protein. In other words, would have a dominant effect. So this is going to be our choice. We were told that there are two correct answers, but let's also check the last one. E, splice site mutation affecting 29 base pairs exon near the beginning of the gene encoding an enzyme. Now let's return to this picture of the double-stranded DNA of the gene, and actually gene consists of two parts. Those parts which would be translated into the protein and those which are not going to be translated into the protein. So those parts which would be translated we call exons, and those which are not going to be translated, I show them with blue color, we call introns. This question might have been tricky, but splice site mutation often leads to skipping the affected exon or mis-splicing. This in turn will lead to a frame shift if the affected exon is not divisible by 3. Frame shift mutation. And the net effect is likely would be a loss of expression, loss of function of the protein, because it's again going to be, such mutation would resemble premature stop codon. So would produce short protein which are going to be non-functional. So net effect most likely would be a loss of expression or a loss of function. This could be dominant, but most likely loss of expression will be recessive unless there is a dosage effect. So because we have to choose only 2 variants out of these 5 variants, this is going to be B and D. And also I want you to pay attention that there going to be a loss of 29 base pairs. If the loss would be of the 30 base pairs, this means the protein is going to just lose 10 amino acids. Usual size of the protein is about 1000 amino acids, so loss of 10 amino acids usually not going to affect protein. Sometimes even loss of 1 amino acid may affect, but usually if it is not in active site, it might not have that drastic effect. But because we have here 29 base pairs, that means that the whole reading frame would be affected. So that means that probably within 20 base pairs after such mutation, we are going to find a stop codon, and we are going to see the same picture as I described before, non-functional protein. And this is going to be considered like a recessive allele, which is non-functional. And this is all for today. Subscribe and see you in the next video. Goodbye.